second webinar in our CADASIL Connection speaker series. Today's presentation is entitled The NIH CADASIL Disease Discovery Program, a clinical and basic research investigation to better understand the disease. Our presentation will be given by the principal investigator, Dr. Manfred Boehm, and two members of the study team. I am Jane Gunther. I will be the moderator. The devastation that CADASIL has on families led a group of volunteers in 2012 to establish the cureCADASIL. Thank you for joining cureCADASIL Association's second webinar in our CADASIL Connection speaker series. Today's presentation is entitled The NIH CADASIL Disease Discovery Program, a clinical and basic research investigation to better understand the disease. Our presentation will be given by the principal investigator, Dr. Manfred Boehm, and two members of the study team. I am Jane Gunther, and I will be the moderator. The devastation that CADASIL has on families led a group of volunteers in 2012 to establish the cureCADASIL Association, whose mission encompasses patient advocacy, education, and research support. The dedication and passion of these volunteers to cureCADASIL has led to a number of successes. Most recently, our funding activities has supported research resulting in identification of a potential therapeutic and biomarkers published this year by the Dr. Joe Abaleda Vasquez lab. By establishing a family registry, we are helping to position CADASIL research into translation to the clinic. A current initiative is establishing a patient sample biorepository, which will support biomarker development that is crucial for progressing therapeutic development. Before we begin today, I would like to go over a few housekeeping items. We've taken a screenshot of an example of the attendee interface. You should see something that looks like this on your own computer desktop in the upper right corner. If you were prompted to select your audio and the default setting is computer audio. If you prefer to join over the phone, select telephone in the audio pane, and the dial-in information will be displayed. You have all joined this webinar in listen-only mode. All callers are automatically muted. At the end of the presentation, members of the CADASIL Disease Discovery team will answer questions that were pre-submitted during the webinar registration process. However, at any time during the webinar, we invite you to type questions in the question pane of the control panel. These questions will be forwarded to the panelists following the webinar. Even though questions will not be answered today, those that are entered in during the webinar will not be answered today, please know that it is of value to know the types of questions the CADASIL community has about their disease and the NIH study. Today's presentation is being recorded and will be available on the cureCADASIL website. The CADASIL Connection speaker series was created to encourage communication and collaboration in the CADASIL community at a time where we are seeing years of research from a dedicated few come to fruition. I thank the other cureCADASIL Research Committee members, Sandra Talbert, cureCADASIL President Nancy Maurer, and our speakers for helping to make this series happen. Our goal is for this to be a unique opportunity for the entire CADASIL community, researchers, clinicians, and families to learn about the current state of research in CADASIL. Our hope is this series will lead to identification in gaps in knowledge and identify ways to bridge these gaps, to accelerate research and create collaborations that will make meaningful differences in the lives of those with CADASIL. cureCADASIL sincerely appreciates this opportunity to have Dr. Boehm and members of the CADASIL Disease Discovery team present their work on this NIH-funded natural history study. Dr. Boehm is Senior Investigator at the National Heart, Lung, and Blood Institute and Principal Investigator of the CADASIL Disease Discovery Clinical Study that initiated in June of 2016. In addition to Dr. Boehm, two members of the study team will be presenting today. Dr. Alessandra Rosario is a staff clinician in cardiology at NHLBI, and Dr. Camilo Torra is staff clinician in neurology at the National Human Genome Institute and they will be presenting their work on the disease discovery study. During the question and answer session, we will also hear from Seth, a patient participant on the study. I am pleased to introduce Dr. Manfred Boehm. Dr. Boehm received his MD from the University of Heidelberg and was a research fellow at the Max Delbruck Center and the University of Michigan. Dr. Boehm began his work at the National Heart, Lung, and Blood Institute in 1999 as a research fellow and continued in 2003 as an investigator. He received a Foundation for Advanced Education in the Sciences Award for Research Excellence from the NIH. Dr. Boehm's lab focuses on the identification of molecular mechanisms of human vascular diseases and new therapeutic approaches for rare monogenic vascular diseases, such as CADASIL. Dr. Boehm's lab established the patient-centered vascular translational program through which they are investigating CADASIL, among other diseases. Please welcome Drs. Boehm, Roferio, and Toro. We are switching over to our presenter slides.
You're muted. Okay. So what do I need to do?
There we go. We have audio.
Okay, Lisa, you are now unmuted.
Okay. So
it
now
looks like I don't have your slides up, though. We don't see you as a panelist, so I will have our backup slides being shown.
1 second.
We should be set.
Are we all set?
Great, you are all set.
Thank
okay.
you.
So this is Manfred Boehm. First, I would like to thank Jane for the wonderful introduction, and I would also welcome and thank all the listeners from the CADASIL community outside here who are listening to our webinar today. I would like to start telling you about what you will hear during this 45-minute talk. I will give you an introduction about the NIH, what it is, the NIH, what is the center of the NIH. Then I will go into the research team. So as you know, you need, it takes a village actually to raise a child, and the team is quite large. And then I will also talk very briefly about our research. The second part will be on cardiovascular evaluation. This will be done by Dr. Roferio. And then the third part will be on neurological and genetic assessment, will be done by Dr. Toro. And in the last part, we will hear about one of our first patients in the CADASIL program, Seth, what he thinks about the NIH and some of the impressions he got. Can you hear me well? Hopefully. Okay, so this is the National Institute of Health Clinical Center. It is a quite unique place. It is the largest research hospital in the world, and it's the center of the NIH Intramural Research Institute. The NIH Clinical Center is a very special hospital. It has 234 inpatient beds. It has day hospitals and outpatient facility, and it runs clinical research protocol. At the moment, we have about 1,800 protocols running at the NIH Clinical Center. This is one fraction of the overall protocols that are over 7,000 that were initiated at the NIH Clinical Center. You find more than 4,000 clinical researchers at the NIH Clinical Center. And what is very important to know is that all patients that are going to the NIH Clinical Center are on a clinical protocol. So the main emphasis of the NIH Clinical Center is to study the pathophysiology of diseases, and also to come up with first-in-human clinical trials. And one particular emphasis of the NIH Clinical Center is the study of patients with rare diseases. Think about that more than 20 million people in the United States have a rare disease. As I said, it's a very special hospital, the NIH Clinical Center. The primary mission is science. And all patients that are coming to the NIH Clinical Center are volunteers on a research protocol. And they are partners and are invested in the clinical research process. Care-driven, the research protocol are care-driven through protocol-centric care. And very importantly, the care is free at the NIH Research Center. We have patients from all over the United States and, in fact, from all over the world. And the NIH Clinical Center is the laboratory, the clinical laboratory for the clinical research at the NIH. So I tell a little bit more about the protocols that we run at the NIH. So all patients that are participating in these clinical protocols must meet strict criteria to be able to enroll. The clinical center itself is not a full-service hospital. It has, for example, not an emergency room. There is no obstetric services. Its goal is mainly to conduct research on the patients. A routine non-emergent patient care service, not related to the protocol, are not provided, but are nevertheless managed in collaboration with the referring physicians. So here it shows you a link where you can get more information on the NIH Clinical Research Center. So now I'm coming towards CADASIL. As you know, CADASIL stands for Cerebral Autosomal Dominant Arteriopathy with Subcortical Infarcts and Leukencephalopathy. This, as many of you know, is a small vessel disease leading to vascular-related dementia that is caused by a single mutation in a gene called NOTCH3. It is a slow onset disease with a clinical manifestation in the 3rd to 4th decade of life. One of the manifestations are migraine, stroke, memory loss, and dementia. It is a progressive disease with currently no available treatment option. So this is the CADASIL research team. The center of it is the NIH CADASIL patients, and we are collaborating with an investigator from UCLA, Dr. Luisa Ariste, who does basic studies on CADASIL. Then we have my research team who does translational studies, and I tell a little bit later about it. And we have a large group of clinician scientists on that. We have Dr. Proferio from NHLBI, who oversees the cardiovascular studies. We have Dr. Kosel, Mehta, and Amar, all from NHLBI, who do functional vascular studies. We have Dr. Chen from NHLBI, who does CT vascular imaging studies. And we have Dr. Toro from NHGRI and Dr. Khan from NINDS, who do the neurological studies. We also have Dr. Goodman from NIH Clinical Center, who does neuroimaging, and Dr. Fitzgibbon from the Eye Institute on eye studies. We also have Dr. Snow from NIMH, who does psychometric studies. And this team are supported by a large support group consisting of research nurse, nurse practitioner, patient care coordinator, and many others. So I would like to introduce to you the clinical protocol. As you know, only the patients are coming into the NIH Clinical Center are part of a clinical protocol. So we have this CADASIL protocol. It's a natural history protocol where NIH and non-NIH investigators are working together. And we have built in that protocol regular visits at the NIH Clinical Center. The protocol contains clinical evaluation, imaging like MRI, CT, PET, MRI. These are all images to look at affected organs and tissue and also blood vessels affected by CADASIL. And we also have a large group of medical consultation consisting of neuro, endovascular, eye, and so on. We also combine this clinical evaluation with a scientific evaluation. So the goal is to perform functional genetics, to actually try to understand how does a single mutation in NOTCH3 is causing the very complex disease CADASIL. To do that, we also use disease biomarkers that we are collecting from blood, but also that we are getting from skin. And then we are developing a certain technology called stem cell technology, where we try to duplicate the disease in a dish to study it in much more detail. So the study objectives of our protocol is to examine the molecular disease mechanism and to correlate that with clinical findings and clinical outcome. The secondary objective of our protocol is the establishment of a clinical phenotype variation. So in this slide, you see the inclusion and exclusion criteria. So the inclusion criteria are any male or female in the age of 18 to 100 years. And what is important, we ask for an established diagnosis of CADASIL determined by genetic testing in the early stages of the disease, in the first 5 years after diagnosis, with mild or no cognitive impairment. Sorry. The exclusion criteria are subjects who are not able to give informed consent, or subjects who decline to provide certain samples, pregnant women, but also subjects, for example, who are unable to undergo an MRI scan. So now switching gears and talk a little bit about the research that we are doing. As I said, and was introduced by Jane, my group works on functional genomics in rare Mendelian vascular diseases. These are inherited vascular diseases, and our center is the patient. So we use the patients to understand the disease, to identify the underlying mechanism, and then move forward and try to establish treatment strategies for these rare diseases. So this shows you our program. As I said, the center of this program is the patient. So these patients comes to the clinical center where we perform the clinical evaluation, as I indicated in all the members of our team. And then we collect biomarkers to identify disease-linked biomarkers. And then we try to model this very complex disease that manifests in the blood vessel in the brain in a very specific way by using and developing patient-specific in vitro disease model system. And as I said, we use stem cell technology. This is called iPS cell technology, or induced pluripotent stem cells, that we use to model certain tissue so we can study it in a patient-specific way. We also use primary cells from the patients, like blood cells or skin cells. So this shows you what we do, what I mean by the disease in a dish. So we have here the patients, and we get some cells from the patients. It can be from the skin, it can also be from the urine, and it can also be from the blood. And we use these cells to make stem cells from these. These are very primitive cells that can be, very importantly, genetically changed. So we can change the cell, we can change the gene. For example, in the dish, we can rescue or repair the genetic defect and see how the cell is changing. And then we can look how these cells behave. So our program is focused on something called mesoderm. Mesoderm is a certain cell lineage. They make certain parts of the body, like the heart, the kidney, muscle, but also blood cells. And as you know, CADASIL, the main clinical problem is a vascular problem. So something wrong with the blood cells, with the blood vessels. So we use these cells to model the disease, but also to use it to treat or to develop new treatment strategies. And we also use these models to actually do drug screening, to find out what kind of drug interfere with certain changes in the cell. So this shows you one of an illustration of our in vitro disease models. Here you can see stem cells, the induced pluripotent stem cells. These are probably, I would say, 100 stem cells, very compact here, that we generate from each patient. And then we use these stem cells to change them into a cell population that is CD31, CD34. These are all markers for a certain progenitor cell population that then will be used to further make then the specific cells we want to make. So we can make vessel lumen cells. These are the endothelial cells, for example, or the vessel wall cells, the smooth muscle cells. And actually the mutation of NOTCH3 is mostly affecting the function of these cells, but we can also make fibroblasts, these are connective tissue cells, or circulating blood cells. Then, as I said, we not only develop these stem cell technology, we also use the patient's own skin cells to look for changes. So, for example, we do skin biopsies, as you can see here, and then we use these skin biopsy for histological evaluation over here, but also for electron microscopic changes. And as you know that although NOTCH3 is causing a vascular disease in the brain, as you can see here, NOTCH3 is shown by a brown staining in the brain. NOTCH3 can be found in a variety of other tissue, including the skin over here, but also in the heart, as you can see here. So this is a blood vessel in the heart. You see the medial layer, the blood vessel wall, that is comprised out of smooth muscle cells that are highly expressing NOTCH3. And oh, I'm sorry. Can I go back? Yeah. And what we are also planning to do is to look into very detailed how these blood vessels are composed and are showing itself by using electron microscopy. And I'm now giving over the presentation to my colleague, Dr. Alessandra Porferio, who is a cardiologist here at NHLBI and who is also part of the cardiology consult service, who will tell more about the cardiovascular studies we are doing on our CADASIL patients.
Hi, thank you, Manfred, for the introduction. As Dr. Boehm was saying, the notch tree is still expressed in the brain, but we do know that it's expressed throughout the entire vasculature. Though it is unclear what is the extent based on the studies that we have in the literature and what are the clinical implications of this expression in the vessels. Because of that, each subject that we enrolled in the CADASIL disease discovery undergoes a series of tests. The neurological testing, specific for the CADASIL, all the biospecimen collections for Dr. Boehm, but they also undergo vascular examinations. Here, I put that table from our protocol to give you a broad view of all the testing that happened at the different visits, time 0, and then after 1 year, and at the end of the study, after 1 more year. When we look at this, all these studies, for us it's very important to start with a full medical history and with a full physical exam to understand each single patient in its integrity with all the medical conditions that can be associated. And we do that through the history, through the physical exam, but also through clinical blood work, looking at kidney functions, looking at liver function, inflammatory markers. Then, of course, are the biospecimens, as I said, and the neurological testing, but we do some very specific just a second. Some very specific vascular testing. We obtain CT angiogram, endopath, cardio-ankle vascular index, and pulse wave velocity. I will briefly go through this testing, starting from the CT angiogram. That is really a clinical test, but we use to answer research questions. With the CTA, we are doing an anatomic evaluation of the vessels, looking both at the lumen, at the vessel wall, and we are extending it all the way from the neck down into the legs. We are trying to understand if any anatomic variant may be present specific to the CADASIL, or if any anatomic variation may be related to other vascular comorbidities. The other vascular test that we obtain is the endopath. It is an FDA-approved, non-invasive test. It provides a measure of endothelial function. It is a very consistent method because it's highly automated and is operator independent. And it gives us an evaluation of mostly the small vessels. Through the CAVI, we evaluate mostly large vessels. We are looking at vessel arterial stiffness. The test is again non-invasive and consists of blood pressure measurements in 4 extremities while we are monitoring the electrocardiogram. Last but not least is the pulse wave velocity that we obtain through the sphygmocore. This provides us an estimate of large blood vessel elasticity and blood pressure close to the heart. It's again an FDA-approved test. It's non-invasive, and what it does uses a blood pressure cuff and a small probe to determine the speed of propagation of the blood through the blood vessels, comparing it then to a control population. In summary, we try to do a full cardiovascular evaluation of our patients. to obtain a cardiovascular risk stratification. We use tests that are looking at both the large and the small vessel, although we primarily know that for now that the CADASIL is involving the small vessel of the brain, because we are looking for any other possible anatomical abnormality. We are looking for endothelial dysfunction, for arterial stiffness and arterial elasticity. This is like a starting point into this protocol. Based on the results of all these vascular studies that we are obtaining, we may consider for the future protocols that are going a little bit more in depth into the study of the vessels throughout the body, and or specifically in the areas that we find to be already involved. This was just a short introduction on our vascular evaluation, and I will introduce now Dr. Toro to continue the presentation on the neurological manifestation of the disease.
Thank you, Alessandra. So I'm going to take this opportunity to present CADASIL from the neurological perspective, and as we proceed in explaining the way CADASIL symptoms emerge, then we are going to introduce in a relatively simple way some of the genetic underpinnings of CADASIL, primarily because we're limited in time and is a fairly a complex topic, but we'll use some I'll use some strategies to convey the idea. So CADASIL, of course, you all are aware of the acronym, and I would say that this particular slide, in a way, sort of embodies the definition of CADASIL. That, of course, CADASIL is defined as a disease of the brain, but as you already know, and as we are trying to study more detail, it's probably a disease of the entire system. It just so happens that the blood vessels of the brain turn out to be the most susceptible to the pathology of CADASIL. CADASIL is an autosomal dominant disease, and that refers to the inheritance of CADASIL, and I will spend a little bit of time towards the end basically delving into the issue of the inheritance. We say that CADASIL is an arteriopathy, meaning that it tends to affect the arteries throughout the system, and it leads to subcortical infarcts that are represented in this slide by that little cavity that you see in the MRI that people refer as a lacunar stroke. That is the subcortical because it's deep inside the brain, and then is associated with leukoencephalopathy, that is the white matter changes that are represented more towards the periphery. So that is a good way of sort of envisioning CADASIL from all its different perspectives. What I'm going to do is actually show you one patient. And this was a patient we recently had the opportunity to see. And again, very important to keep in mind, every patient with CADASIL is different. So when we look at the statistics and we see incidence of these or incidence of strokes, incidence of migraine, keep in mind that those are aggregates, and that in a way every patient is going to have his or her own particular presentation. So I'm going to show you the images of a woman in her 40s, fully productive, employed, highly educated, and has had a very active life, and approximately at the age of 45, suddenly developed weakness of the left side of the body. She went to the emergency room and they did a CAT scan, and I'm showing you the images of the CAT scan here. The CAT scan didn't show anything that would be typical for a stroke, but did show some unusual changes in the intensity of the white matter. that wouldn't definitely be a good explanation for her symptoms of left-sided weakness. This was followed by an MRI, and the MRI actually was very definitive in showing a particular area that I'm showing you right there on the right side that is consistent with an acute stroke. This slide changed too quickly, and I don't want to go backwards. But that area with that high intensity basically tells you that there is an area that is undergoing acute injury due to stroke. That image could be repeated a few months later, and that area disappears, telling you that the acute stroke is already subsided, but usually left with an area of permanent injury. What was unusual about our patient was the presence of these white matter changes, and they are represented here on the MRI, and that is the leukoencephalopathy part of the MRI. So our patient had that very unusual sort of an acute stroke in the context of an incidental white matter abnormality, and the combination of the two, plus the fact that the family history suggested the presence of early onset strokes in her mother, were then good hints to suggest that CADASIL was the proper diagnosis, and this subsequently was confirmed by the proper testing. This would be another patient. This is a patient who, again, presented with left-sided weakness in the emergency room setting, has those changes of an acute stroke, and then on a subsequent MRI, you can see the CADASIL-related white matter changes. When one looks at the MRI of patients with CADASIL, there is an abnormality that tends to be fairly distinct, and is the presence of white matter changes in the tips of the temporal lobes. Those changes are actually fairly unique to CADASIL. There are many diseases that produce white matter abnormalities, but CADASIL would be probably the one that would be distinctly associated with those abnormalities of the temporal lobes. Interesting aspect of CADASIL is that we are talking about strokes, we are talking about blood vessels, but if you look at the blood vessels of the brain with a magnetic resonance angiogram, you don't see anything that is abnormal. And the reason is primarily because the abnormalities are beyond the resolution of these techniques. The abnormalities tend to be, the abnormalities that lead to stroke are actually probably several divisions after those branches. That the smallest branches that we see here are probably the third divisions, and CADASIL-related strokes tend to occur in blood vessels that are probably in the fifth, sixth, seventh division of the blood vessel, so they will be beyond the resolution of an MRI. When brain tissue is examined in patients with CADASIL, the typical abnormality that is evident is in the blood vessels, and is in very small arteries of the blood vessels of the brain and blood vessels throughout the system, most likely, and are changes that affect the media of the blood vessels. The blood vessels have three main layers, and the media layer is very important. It has this small muscle cells, smooth muscle cells, and CADASIL basically produces degeneration of that layer, and that degeneration over time actually has a tendency to induce basically a stroke that is confined to the area of damage. When one looks at the specimens, this will be an autopsy specimen. What you tend to see is actually you tend to see the areas of stroke, for example here, but then in the areas where you see a lot of the white matter changes, what you tend to see is this very small dilatation of the perivascular spaces, and that can be seen in the microscope. So when one looks at the white matter changes are really representing these dilatations of the perivascular spaces. So CADASIL, again, this was covered already, occurs usually in as adults, leads to strokes, cognitive issues. There's a history of migraines, not in everybody. There could be symptoms of depression, apathy, and is due to mutations in the gene NOTCH3 in at least 95% of the cases. So this leads us to the discussion of the genetics of CADASIL. So let me just define a few terms and use a very simplistic, and I really apologize if many of you see my presentation as too simplistic, but I love to use the analogy of our genome, our compilation of all our instructions as an encyclopedia that contains all the recipes to make the components of our body. So our genome is comprised of actually about 3 billion letters. And those letters, so you can think of the encyclopedia of recipes as containing a total of about 3 billion characters. And in those 3 billion characters, we have actually the instructions to build all the proteins in our body. And we have, humans have approximately 20,000 proteins, different proteins. So we can say the encyclopedia contains the instructions, the recipes for about 20,000 different proteins. And those recipes basically are determined by the order in which these building blocks called amino acids are put together. So if we talk about NOTCH3 by itself has 2,322 amino acids. And the sequence in which these 20 basic amino acids are put together leads to the particular characteristics of NOTCH3. So what is a mutation? And in particular, what is a mutation that would lead to CADASIL? So a mutation would be really like a spelling error in our food encyclopedia. And it's a spelling error that just happened to change the order of the letters and leads to a change in one of the amino acids. And there are particular amino acids that are more important than others, and we can talk about that in a second. Now, there are different mutation types, and this is important because for CADASIL, it turns out that all the mutations are what are called germline mutations, as far as we know. Germline mutations mean that those are mutations that exist in every single cell in our body and that are transmitted from parent to child, as opposed to what are called somatic mutations that wouldn't be pertinent for CADASIL as far as we know, that implies changes in the genetic code that emerge after conception. So, for example, this would be very typical for cancer, where, for example, if you develop melanoma, very likely you develop mutations in the DNA in cells of the skin. And those are mutations that are unique to a particular tissue, for example, the skin in melanoma, and are mutations that are not transmitted from one generation to the next. So in the case of CADASIL, germline mutations are the ones that are the most, relevant. And then we talk about the inheritance pattern. The inheritance pattern could be dominant or recessive. Dominant means that that only one mutation is required to develop the disease, and recessive means that both copies of your gene, remember, and I'll go through this in a second, each one of us has 2 copies of each of the 20,000 genes that code for protein. There is a copy that is provided by the mother, and there is a copy that is provided by the father. And in the dominant diseases, just one bad copy may be enough to give you the disease, whereas in recessive diseases, you have to have both copies having a problem for leading to disease. So let's assume that we are going to dig down into the genetics of CADASIL. So our DNA resides in the nucleus of the cell. In that nucleus of the cell, we have the chromosomes, and the chromosomes are arranged in pairs. And you see the yellow chromosome belongs to the mom, and the blue chromosome belongs to the dad, so the 2 copies. For CADASIL, the mutation is actually in chromosome 19. So if we dig in chromosome 19 and use like a GPS, that GPS will take us to the location of CADASIL that is in this very, very unique region of chromosome 19. Okay. The genes are packed in little packages that are called exons, and if we unpack those exons, we actually get to the genetic code. And if we change any of these letters in the genetic code, for example, here, we change the letter C, we can change this cysteine to a different amino acid, and that may be enough to actually give us CADASIL. It turns out that CADASIL, the notch mutation in CADASIL leads to a change in a very particular part of the protein. It turns out that the protein notch 3 has a portion that lays outside the cell and a portion that lays inside the cell. And our mutations only on the outside of the cell in a portion called the EGFR, the endothelial growth factor receptor portion of the protein that is capable of leading to CADASIL. And there are 34 domains that repeat themselves, and any of those 34, if they have changes that affect the configuration of that EGFR domain, that could be enough to lead to the disease. In our cohort, and in our patient participant group, we have seen currently a total of 12 patients. And as you see, there's something very unique about CADASIL is that the great majority of the patients, the mutations affect one particular amino acid, that is the amino acid cysteine, that is represented here with the red letters. Either the mutation will take a cysteine out or will introduce a cysteine in, and that leads to changes in the charge properties of the protein that are enough to distort the protein and lead to the dysfunction responsible for CADASIL. Now, the family history in the patient I first introduced was very unique in that, as you remember, I presented to you the patient having a stroke at the age of 45. It turns out that her mother had had a stroke early on in life, and as a result, when taken in combination with her history of early onset strokes, it became very suspicious for a condition such as CADASIL, and that led to the genetic testing. So in this way, I have covered in relatively fast pace the neurological basis of CADASIL and the sort of the genetic principles that determine how CADASIL is inherited and how it emerges as a result of a change in a simple letter in the human genome. So I look forward to your questions, and thank you very much. I'm going to introduce Seth. Seth is a patient in our program, and he's going to talk to us about his experience as a person being diagnosed with CADASIL. And Seth, thank you so much for being part of the meeting today.
You're welcome. Well, I was diagnosed with CADASIL, I think about 18 months ago. Members of my family have been afflicted with it, so I'm very familiar with it. I still haven't ever had any symptoms. But while my wife was pregnant, we talked to a genetic counselor and expressed a concern about the possibility of passing on CADASIL, and the doctor ordered the genetic test on me, and it came back positive, which was a big disappointment to discover. And as I said, having family members afflicted with this illness, I know exactly what it does. Subsequently, my wife saw an announcement, I think in a Facebook group, about this study. So I contacted NIH and offered to participate. I was asked some screening questions over the phone, as I recall, and maybe also further written questions. And eventually was scheduled to come up to NIH. I'm local. And I think I spent 3 days here about a year ago going through quite a lot of tests. I met with the research team. They explained to me what each of their roles were in the team, what kind of tests they were going to do, and what they're studying and trying to determine. There's a lot of patients involved in being a test participant here. You have there's a patient coordinator who gives you your schedule and makes sure you know where to find everything. But you do. It is a hospital essentially. You go and you know wait in a waiting room until you're ready to be seen. And I think I've had my blood pressure taken on all 4 of my limbs several times. I've had an MRI and a CT scan last year. I had an MRI earlier today. And you know that you just lie still for a while. I did have an eye exam that was rather unpleasant because the dye made me nauseous. Otherwise, I would say everything else is just, like I said, you just lie still or be patient as the doctor or other medical professional is doing their test on you. It's very educational and informative. I do have white matter lesions on my brain, so while I don't exhibit or experience any of the symptoms that my mother or my sister, for example, had for years prior to their diagnosis or since, it is clear that the illness has begun a progression. Consequently, I now see a neurologist. There is still no treatment for this illness, so we just talk about staying in generally good health and monitoring my condition. Dave, do you have questions or things that you'd like me to address?
No. Tell us just a little bit about the family history. I think.
Sure. So my mother, my sister, and one of my mother's brothers is afflicted with CADASIL, or was. Mom had neurological symptoms. I was a kid at the time, but I think she was in her 30s, and she was misdiagnosed with MS for a few years. She would have migraine headaches for as long as I could recall. She complained of some neurological symptoms that were unpleasant. She said she always had this bad taste in her mouth, or a pins and needles sensation, like on her left hand, as if it had fallen asleep, like she'd, laid on it. My sister also was afflicted with migraines, and my uncle was also afflicted with migraines. My sister had a stroke one day. I think she was 30. She was at my house at the time. I did not know what to do with her. We did eventually sort out that we needed to take her to the emergency room. They were very confused by the presence of a 30-year-old exhibiting stroke symptoms, and even more confused when she started to improve. It happened again a few years later, and it was at that point that the neurologist seeing her at the hospital tracked down CADASIL as a possibility. He ordered the genetic test, and of course it came back positive. My sister and my mom compared notes and realized that this sounded also like what my mom had, and so she was tested subsequently also as my uncle. The story gets pretty bleak from there, honestly. My uncle died, I think, about 10 years ago. My mother died earlier this year. My mother was, in full-blown dementia in her nursing home the last 3 years of her life. My sister, who's young, she's only 47, is also in full-blown dementia and in a nursing home. I'm 44 and, still maintaining normal activities, hold a job, and Yeah. So, but obviously that's not going to last. I don't know how.
Any other questions? Any from staff or?
Yeah. Well,
thank you.
Well, we appreciate you participating, Seth. You and all the patients that have, come to NIH and been patient and undergone, the test.
It does require patience.
It does.
This defines patience in a sense that, there is the idea is collect the information in everybody the same way, under the same protocol, so that the information is comparable across individuals. So it introduces a level of sort of monotony to some degree to some of the process, but every data, every piece of information hopefully will help us move forward the understanding of this disorder.
So I think now we can conclude our presentation, and we would be happy to answer any questions either from the CADASIL Foundation as well from any viewers or listeners of our webinar. Thank you. What do we need to do?
Thank you, Drs. Boehm, Porfirio, and Toro, and Seth for a very interesting presentation, very informative. We now are going to go into our questions that were sent to us during the registration period. And our first question is from Savannah. Have you discovered common comorbidities in patients with CADASIL, for example, small fiber neuropathy or cognitive decline in the absence of TIAs?
This is Dr. Toro, and I'm going to answer this question. We have not systematically looked at the issue of small fiber neuropathy, but we have looked at other aspects of CADASIL. For example, we have systematically looked at the network of blood vessels around the retina, and some people think that looking at the back of the eye is like having like a pinhole view of the inside of the brain, and it appears that there are some changes in the vascular network of the retina that could be common to many of these patients. So that's an aspect that is outside of just having the sort of the typical presentation of CADASIL. There's also this suggestion, and it's something that we are very interested in pursuing, that there are changes also in the way the blood vessels throughout the body are responding to blood pressure regulation and to sort of the way the studies that Dr. Porfirio outlined, that there are changes that could represent evidence of the entity CADASIL sort of manifesting in other parts of the body, including the skin and the regulation of blood flow to the extremities. In regards to cognitive issues, our neuropsychologist, Dr. Joe Snow, has conducted a very extensive battery of cognitive testing in all our patients, and there are some themes that are emerging, but I would say that everybody is a little different, and we are looking forward to looking at our patients over an extended period of time to generate more definite conclusions.
Thank you.
I don't know if the rest of the team has any other thoughts or comments?
No.
Okay.
Thank you. Our next question is from Phyllis, and is, should blood thinner be used if a stroke occurs?
That is again, Dr. Toro, and this is actually a very crucial question in the care of patients with CADASIL. And of course, the question even should be framed as, would a blood thinner work in a patient with CADASIL? the strong blood thinners like TPA, that is a clot buster, or coumadin or heparin, that are sort of the stronger blood thinners, are primarily intended to treat, in the context of stroke, I should say, are primarily intended to treat what are called embolic strokes. Those are strokes that, you know, a large blood clot forms in the heart or in the arteries in the neck and then flows downstream and blocks a smaller blood vessel. In that context, one can say blood thinners could be indicated, but CADASIL is a totally different entity in itself, in that the changes are not the results of a blood clot that has become loose and traveled downstream, but rather a process that is occurring at the blood vessel in place, in situ, and that most likely is associated with clumping of platelets where the blood vessel is locally damaged, and that clumping of blood vessels produces the stroke. And the stroke typically is at a very, very distal, kind of terminal blood vessel, very far away in the periphery. And in those cases, most likely blood thinner like TPA or heparin or coumadin would not be beneficial and actually have the potential risk of inducing a hemorrhage. So the answer would be conventional blood thinners should not be used in a stroke that is directly related to CADASIL. I don't know if we should expand any further. Let me know.
Okay. Thank you. I think that is a very good explanation. I think that has been a question that certainly many people have wondered about, in terms of, you know it's a disease of stroke, so are other stroke drugs appropriate? And I think it was a very good explanation. Thank you. Our next question is from Jane S. Are there any vitamins or supplements or diet to help with CADASIL? Maintaining optimal brain health seems important. Are there thoughts from the panel on what a patient could do on their own, or in help with their clinician?
Dr. Bolseghi, I'm going to answer this question from a cardiovascular standpoint, because from a CADASIL standpoint, really we do not have proof that any supplement, any vitamin would improve the health of the brain. We always suggest all the patients that are coming through with CADASIL to maintain a very good cardiovascular risk factor profile, so trying to avoid to develop all those kind of diseases that could further damage the brain through the process of atherosclerosis, so like a healthy diet and regular exercise, but no specific supplement or diet supplement that is for the CADASIL itself. So anything that a cardiologist will tell you to keep the amount of fat down, not to eat too many sugar drinks, to keep your weight in the normal range, and to keep your blood pressure in the normal range. Those are all the important factors to preserve the brain, to try to avoid any further deterioration.
Thank you. Is there a component of the study that helps to identify when a CADASIL patient's cognitive function is impaired to the point that they can no longer do normal job responsibilities, such as keeping a timeline or making decisions, and the amount of time that a person without CADASIL would be able to respond?
This is Camilo Toro again. So I would say that we, through the neuropsychometric testing Dr. Snow completes, he is able to dissect with high-level precision sort of the cognitive function of each individual. With respect to particular tasks and functions and risks, it becomes a very, very individualized decision that is probably not a decision that is best made in the context of a clinical trial, that is probably best determined kind of at a one-to-one interaction with the local clinician, for example, in relation to driving or to other responsibilities involved in decision making, that is something that probably would be best approached with, for example, for driving would be through an occupational therapy assessment or a behind the wheel assessment, because there could be patients that CADASIL may affect them differently, and you don't want to really use like a one-size-fits-all to all patients, and that this really has to be individualized.
Okay. Is the type of neuropsychometric testing that is being done on the study, I'm assuming that's something that is not readily available for patients that are not near the NIH. Is that correct, or is that something that major cities you would be able to find someone who could do that type of testing?
This testing is done by a neuropsychologist, and is I'm sure that a neuropsychologist will be able to perform it at a different place. It's fairly involved. It would, and Seth can help us. How long did it take you to complete your neuropsychometric testing?
It was something on the order of 2 hours. Yeah. You know, it was a lot of, well, it ran the range of following simple instructions to completing logic puzzles and memorizing strings of numbers and that kind of thing.
So it is
taxing.
It's taxing, yes. And this testing has been very, very carefully. This is not necessarily defined for CADASIL patients. It's a battery that Dr. Snow, so he has a battery where he does a general initial screening, and then he's sort of, depending on the profile of each patient, he may kind of dig in further into certain different domains. So it's conceivable that the neuropsychometric testing that Seth had may be somewhat different than the neuropsychometric testing the next patient would have, based on the initial profile. And then if an area is identified that has, say, for example, a particular weakness, then Dr. Snow, at the return visit, will kind of zoom in into that area and dig further to see, is there any suggestion that this was, is this changing, is this stable, does it need any additional testing? So I would say that is more intended to get a very detailed and very comprehensive view of cognitive functioning, rather than generating sort of actionable data to make decisions related to activities of daily living. That would be kind of the way I would frame it.
Okay, thank you. Low milligram aspirin, baby aspirin, is recommended by some CADASIL patients' physicians for their patients. Are the people on your study on any medications for CADASIL like low milligram aspirin? And could data from this current study provide an early read on whether baby aspirin is safe and or effective for CADASIL patients?
Okay. Low dose baby aspirin, we are recommending our patients, and I think we have recommended every single one of our patients to go on low dose baby aspirin. That would be 80 milligrams daily. Of course, one would have to consider if the patients have any particular associated risks to go on aspirin, for example, history of bleeding or history of gastric ulcers or allergy to aspirin. But we are recommending in our patients the use of baby aspirin. Now, that recommendation is based primarily in sort of the anecdotal and sort of standard of care of CADASIL patients across the community, not only here in the US, but across Europe. But the data on the efficacy and safety of it, I think there are no really good systematic studies that have answered that question. I think, unfortunately, our study is not powered, doesn't have the high number of patients that will be required to be able to answer the question whether aspirin will be safe and or effective. That would require more of a multinational effort and And that actually would be a pretty important but pretty difficult study to complete.
Thank you. That's very helpful. Our final question is, how many participants are included in the current study? Is there funding for more participants in the future?
Yeah, so this is Manfred Boehm. Currently, our protocol is set to, our study is set to work together with up to 20 patients. This is a very small number compared to the overall CADASIL community. However, keep in mind that we really perform very detailed clinically, but also research evaluation. That would be difficult to do on a larger set of patients. But we do have possibilities in place to enroll more patients through slightly different protocols. If we find something in these initial 20 patients that we would like to follow up. And we also, so this study is a clinical protocol. It's funded through an NIH, NHLBI, UO1 grant. And we are looking forward to further funding to expand this study to look for different disease mechanism opportunity findings and how we can translate that into potential treatment strategy. Yes, so funding is essential even here at the NIH, and yes, we are looking to obtain more fundings, yes. Thank you.
Thank you, Dr. Boehm. That was our last question. I would like to thank our speakers today. It was very informative, and thank you for the work that you are doing to really find a treatment for CADASIL. In closing, you can join us at our next webinars. Our goal is to connect the CADASIL community. Please contact cureCADASIL at gmail.com if you would like more information from our speakers or if you have a topic of interest. Recording of today's webinar will be available at our website. Thank you to today's speakers for their work advancing understanding of CADASIL and for taking the time to share their work with us. Thank you for your audience participation. This is the conclusion of our webinar.
Thank you.